Okay, so welcome back. We are still at energy of activation. And notice when you look at this figure, there's this delta G and there's that value. Remember that value should be negative. You're going to actually learn, hopefully, why the negative is. If this is uncatalyzed and this is catalyzed, the difference truly is the amount of energy that's required to drive the reaction here. So this unstable state is called a transition state, where what we're doing is, let's take a look here. Let's go A plus B. So our A plus B is over here, plus B. And somehow the electrons between these need to be excited in such a way that they will be able to find the correct orientation so the octet rule is fulfilled between these two sets of reactants. Okay, A is one set, the other is B, such that when they come together, they've fulfilled the octet rule in either some ionic, some covalent bonding structure that will now have the product AB down here, and the amount of energy that was required for that to happen is here. And the whole point, of course, is that that transition state happens really quickly. Okay, and what happens is the heat energy is the main source of activation energy inside of a cell. So that means that depending upon what type of reaction we are doing, it's going to require, if you think about this for just 1 minute, how does your body have a temperature? If you think about the amount of energy that's released into the system as chemical reactions occur, this means you can begin to understand why it is during normal metabolism, with all of the sum of the anabolic and catabolic reactions that are occurring, that transient state release of energy and the activation energy required for chemical reactions to occur allows the heat to help reactants reach that transition state. And this is how you build things, using anabolic reactions. And that energy can then be trapped so that you can now use it to drive catabolic reactions. Now that's a lot, okay? But let's just put this back down here. A plus B. And they give us AB. So this would be an anabolic reaction, right? And it means something is going to be here to allow for that reaction to go forward, and that something either decreases the energy of activation, something along those lines, to make sure that the product here, okay, can actually be formed. Now, what's going to happen, of course, is, where are we here? You're going to notice that it's because it's catalyzed, we actually end up driving this forward. Can you guess what catalysts do to reactions? Well, remember, I've given you this really big introduction in terms of catalysts tend to be metals. Those metals tend to stabilize reactions because of their shape. It means they're holding the reactants in place in a specific orientation. That A is being held in a specific orientation. That B is being held in a specific orientation to allow the likelihood that that transient state or transition state to get us that product will in fact be reached. Okay? And now when you think about it, what's going to happen is the breakdown, of course, is an exergonic reaction, and that's the amount of energy that's going to be released by the breakdown of, in this case, some ethanol/gasoline type product. Not necessarily as important for us, but if we think about what this means in terms of if this was, say, a glucose molecule, okay, once the reaction begins, enough heat is released to drive additional reactions, which means once you take a C6H12O6 structure and you begin to catabolize that in the presence of oxygen, such that you end up making CO2 plus H2O plus ATP. And I want you to see that this is not a balanced reaction. I don't really care about the balanced reaction at this point in time. What I want you to see is that the oxidizing material here is oxygen. Our glucose is going to be broken down. And, you know, what you need to remember is that that C6H12O6 structure doesn't look anything at all like this, which means this is going to be broken apart, which means all of the energy that's being released from this is going to be converted into some intermediate that we're going to know as water, and some of the energy is going to be trapped in phosphate here, between the phosphate bonds. Now, How does this happen? Well, it happens because some people ask the question, well, what are the governing rules that allow all of these reactions to move forward? And at the end of the day, they come down to this really important concept of what are the basic laws of thermodynamics. And thermodynamics is a study of energy and energy transfer involving physical matter. All material in our known universe happens in only two states. It's either free energy form or it's matter state. And the first law of thermodynamics states that the total amount of energy in the universe, if constant, is that energy cannot be created nor destroyed. That also means that matter cannot be created nor destroyed. Okay? And the second law of thermodynamics states that the transfer of energy is not completely efficient, which is great because if it was completely efficient, it would be directed and we wouldn't have free energy to drive reactions. What it means for you is you need to understand that a chemical reaction, some energy is lost in the form that is unusable, such as heat and energy. And that is what's causing an increase in the amount of entropy in your known world. And that's required. I mean, people will say the world is falling apart around you continually, and it is. Okay? The fact of the matter is every day you get up, you make your bed, you reduce the entropy of the universe every day you do that, right? And in doing so, you allow for a little bit more order and a little bit a reduction in the total amount of chaos inside of your nervous system when it looks at a clean bed as opposed to a messy bed. Why do we care about this? Well, because this goes to be these kinds, these kids, oh, I see what they're saying here. These kids convert the chemical energy from the ice cream cone and the sugar sprinkles and the actual cone itself, because that's a sugar cone and not just a wafer cone, right? They're going to convert all of that into riding their little stabilized bicycles with their training wheels here. And notice they have their helmets on, so when they fall off and their heads get smashed into the ground here, it's not going to harm them that much. But by the time they go running to the house going, look, Mommy, look, Daddy, look what I've done. I rode my bicycle. Good for you, kid. Anyway, the point is this, of course, is that if you put your hand on them, they would be warm. Likewise, if you got up and went for a run, you'd find yourself getting warm as well. It is simply a continuation of this idea that light energy from the sun drove the formation here of this material. This material can be converted into something along these lines here. I think those sugar things are all chemicals that are bad for you, but whatever, go eat them. Okay, and then you can run around spastic like a little child. Now, kids are supposed to run around spastically. It's kind of cool when you think about it. One of the interesting things is, of course, is we're supposed to learn how to control all of that free energy, and that heat is also lost when plants utilize sunlight during photosynthesis. And I think what they mean to be telling you here is that the leaves actually absorb not only the light, but also heat, and that heat allows for the driving of photosynthetic reactions inside of plants. Now, how then do we actually get this to be useful for us? How do we actually allow for cells to drive endergonic reactions? Because we need to increase the complexity of some structures. Imagine we went from, say, 1A. Let's go like this. Let's go 1A. Take that 1A, and imagine we wanted to add it to another A. Oops, it's supposed to be an A, Jonathan. And another A here, and another A here. Now, all of this would give us this structure A. 1, 2, 3, 4. 2, 3, 4. And the molecule that they use to do that is they hydrolyze the energy from an ATP molecule, and they transfer that energy into the bonds that they have here. And that's where that energy is added. Okay? So now let's see how it actually happens. Wow. So ATP is a nucleotide, and your textbook doesn't necessarily outright describe it as that, but it is adenosine, which makes up one of the nitrogenous bases from adenine. Okay, it has a 5-carbon sugar attached to it. That's that ribose. Oh, look, we could actually make RNA from this if we wanted to. But the idea, of course, is that 3-phosphate group here, 1, 2, 3, which we've seen, looks like we've seen something like this when we're looking at phospholipids. They actually have 3 of them, where alpha, beta, and gamma, and the gamma phosphate is literally cleaved such that this bond is broken here, and when that bond is broken, the potential energy stored inside here is now released in a really small area, and that really small area drives those endergonic reactions your textbook is asking you to think about. When the bonds are broken, the products have lower free energy. Great, who cares about that? What's important to understand is that this allows for driving those reactions such that the products will have a lower free energy because the energy came from someplace else. Excuse me, sorry. So, takeaway point here is that Mother Nature has created and repurposed what looks like some intermediate structure that we might use for, say, nucleic acids, attached a series of phosphates to them, so that sugar phosphate backbone, do you get it? Sugar phosphate backbone. Okay, could then, because that's a sugar phosphate backbone right there, she accidentally threw a couple of phosphates on here. And if this became labile, and you could break that bond, that meant that anywhere this was someplace around something where energy is required, you broke that bond, that energy would now be made available. And that is interesting engineering. And that's what you actually see here. The interesting thing about this, of course, is here's the number you do have to pay attention to. Notice that ATP is an unstable molecule, and it will hydrolyze quickly. And notice it has a negative value here, which means it generally tends to go towards entropy. Okay, if it's not coupled to an endergonic reaction, their energy is lost as heat. And now just take a minute and place your hand on your neck and ask yourself, what do I feel? And hopefully what you feel is heat, where if ATP is what's driving all of the chemical reactions inside of all of the cells of your body, as well as some frictional heat between the small machines that are actually working inside of your body, what happens is that heat is lost into space, and that space is what you feel. Okay? And the interesting thing is, of course, is that Mother Nature is now going to allow for hydrolysis to be reversible. And so this is a very important reversible type reaction where if you actually remove this phosphate here, you actually end up with ADP plus phosphate plus free energy. But that also means we can take free energy, attach that phosphate back to ADP, and now we have ATP. And this can keep going along as long as these reagents are available for chemical reactions to proceed. So how does this all happen? One example of how we use sodium-potassium pumps is, in this case, we're pushing something against its concentration gradient. So remember, I started this lecture off with making sure you understood that Mother Nature uses diffusion gradients, osmosis, and you remember primary and secondary active transport. Well, what's going to happen is ATP hydrolysis is responsible here for pushing 3 sodium ions out, 2 potassium ions in, because that release of the energy when we cleaved ATP into ADP plus PI. Let me draw that out for you so you understand what I'm saying here. So we start this with a sodium potassium ATPase pump here. ATP, okay, is present. It's going to be cleaved into ADP plus P. And what's going to happen is that free energy is going to move around. Well, free energy here literally changes the shape of this pump. And that pump now allows us to push 3 sodium ions against the concentration gradient of sodium outside of a cell and inside of a cell. So it takes energy to push something against its concentration gradient. Now a change in the shape of, in this case, this transporter now allows potassium to bind here. And when it binds, it then removes the phosphate that was temporarily attached here, and now it goes back to its original shape. And this entire cycle keeps going on so that you maintain what's called a resting membrane potential. And that's what I mean when I say cells that are excitable have a positive charge on the outside of the cell and a negative charge on the inside of the cell, because you have something that's called the sodium-potassium ATPase that establishes and maintains that resting membrane potential. You are alive because ATP can be hydrolyzed, so you can maintain this gradient across the membrane. Now, how do we actually use this? If we think about what enzymes are proteins. These proteins reduce now the formation of products, and that's because these enzymes are very specific, catalyzing a signal reaction. And what this means is Mother Nature built a specific protein to ensure that a specific chemical reaction occurred. And notice the dotted red line here is the reaction with the enzyme, or in this case, the catalyst is a protein catalyst. Okay, they're not always proteins. You can have some ribozymes, which are really important for us to understand in more advanced biochemistry classes. But for right now, I want you to start thinking about that there are subsets of proteins that are enzymes, and those enzymes catalyze chemical reactions. And how do they do it? They do it because the enzyme and reactants, which are the substrates, determine the specificity. And they all get together in an active site, which stabilizes the possibility of some bond being formed here. Okay, and what's going to happen, or in this case, the bond's going to be broken. Okay, what's going to happen here is the enzyme doesn't get burnt out in the process. And what happens is you now have your two unique products. So this would be an example of A being broken down into A plus B. I tend to try and teach this in terms of like, has anybody ever tried to set you up on a date? Okay, so think about the reverse here, right? The matchmaker is the enzyme. They bring the two reactants together and they say, hey, what happens? You guys should go out for a date and find out if you actually get along. And the reality is sometimes it works out and sometimes it doesn't, yada, 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 blah, blah, blah. Okay, but that's what's actually happening here. The enzyme acts as a facilitator to allow for the chemical reaction to occur. Now, this is a specific protein structure. It's a three-dimensional image, and you're going to see I'm going to have you guys go look at some of these a little bit later on. The idea, of course, is that the specific shape is what drives the chemical reaction. So it can bind, hold it in the correct orientation, and then the catalytic site reduces the chemical energy of activation. Okay? And you can see here, this is what's going to bind inside those spaces. They're making this overly complicated just so they can dazzle you with their ability to draw 3D models. But if you took this class, or if you take this class with me in the fall, we're actually going to build these 3D models ourselves. So how does that mean? So what does this actually mean? What I want us to think about is protein structure that is revisited. And remember, the individual personalities of all environments inside of a cell or an enzyme are optimal or suboptimal depending upon temperatures or pHs, which means each enzyme will only be active in that unique temperature, in that unique pH, and in its unique shape. Its three-dimensional shape of the protein will determine by the amino acid sequence of the polypeptide that when it will be functional and when it will not be functional. This is really important if we think about something like a red blood cell, okay, where in one part of your body the pH is X, and in another part of your body the pH is Y. In the area of X it has one responsibility, and in the area of Y it has a completely different responsibility. Okay, so where are we going next with this? So how can they do this? Well, I've already told you how they do this, right? They lower the energy of activation. You can actually see how this works here. I recommend you actually go look at this. This is actually kind of cool. And again, it's just a couple of computer scientists who are just have a little too much time on their hands, and you're looking at how the enzymes actually work. But it's kind of cool. I want to pause here so we can get back to what enzyme regulation looks like in its own video. Okay, so we're going to stop a little bit earlier. I spoke a little bit longer. I'd hope to get all of these things done